So now we will hear from Alex Juncker. Alex joined the lab as a graduate student in 2022 after completing their MPH in social medical sciences here at Columbia. Their PhD work focuses on how ultra-weak photon emissions influence cellular processes, but they also have a MiSBIE project on mitochondrial disease and allostatic load they are going to present about.
Thank you for that introduction, Carolyn, and thank you everyone for being here today. As Carolyn said, my name's Alex, and I'll be talking to you today about the project I've been working on measuring allostatic load in the MiSBIE study. Sorry, thank you. So we're interested in allostatic load primarily because we're interested in stress. So as we all know, psychological stress is not only experienced in the mind, it has profound effects upon the body as well. This is good. This means that we're able to respond to stressors, whether that's through flight or fight, things like that. And to support these stress responses, there are changes in cortisol and catecholamines, glucose, blood pressure, along other markers. As I've said, this is adaptive, this is good. But what's also important is that after the stressor is removed, that these markers go back to their baseline levels. When this doesn't happen, when there's kind of a chronic alteration in these markers, it's termed bodily wear and tear, and this gets quantified as allostatic load. So when I speak about allostatic load, what we're talking about is the kind of long-term elevation or decrease in markers resulting from chronic psychological stress. So this could be extended durations or repeated stressors. Allostatic load itself has been studied for decades at this point. It's been shown that people with high allostatic load are more prone to depression and other mental health conditions, and to neurodegenerative disorders, including Alzheimer's disease. And in addition, there's just a general decrease in physical and cognitive health. So this could be increases in frailty for people with high allostatic load, things like that. This has been shown to be related to interpersonal stressors or chronic stress that's experienced. This has also been shown in large epidemiological cohorts to be related to income or to more structural factors like this. Our lab, of course, is interested partially in why this happens, and with our focus on mitochondria, we're curious about the energetic cost of these repeated adaptations. So every change that I mentioned within the body to respond to stress requires energy, which is mostly provided by mitochondrial oxidative phosphorylation. And previous work in the lab has shown that with energetic stressors caused by genetic mitochondrial defects, there are systemic effects. So this has been shown in cells with mitochondrial defects. These cells use more energy, they age faster, and they have altered cytokine levels. So hard to say this is systemic in a cellular monoculture, but these cytokines would be secreted and measured as something systemic in people. Further, in mice, we've shown that mice with genetic mitochondrial defects, they have an energetic stress that alters the stress response and triggers inflammation as well. You heard about this from Martin earlier in the day. So all of this work together has brought about a model put forward by a previous doctoral student in the lab, Natalia Boba Alves, termed the energetic model of allostatic load. So this proposes that the load in this is really the energetic burden caused by the need for repeated adaptations. So the goal of our project here is to examine whether the energetic stress of mitochondrial disease alters systemic dysregulation in humans. So essentially, does this model translate if we look at this in people, or do we see kind of a refutation on some level? So to do this, I asked three main questions here. First, is allostatic load higher in people with mitochondrial diseases? Secondarily, does it come from the same systems? And then third, how or does allostatic load interact with psychological stress in a number of forms? So we did this in a total of 105 people. Using about 35 different biomarkers, we quantified allostatic load. I did this in a couple ways, which I'm happy to talk about if anyone has questions. And then we assessed stress in a couple ways using different questionnaires, which I'll go into later. But here, starting with the first question about whether allostatic load is higher in people with mitochondrial diseases, we can very easily say the answer is yes. We see this as statistically significant and with a pretty moderate effect size that people with mitochondrial disease do have higher allostatic load. We looked as well at the correlation of allostatic load with age. In controls, this is statistically significant. There's a strong correlation as we would expect there to be. However, in the group with mitochondrial disease, we see that these matters are not correlated. This is kind of surprising, but at the same time, it recapitulates what we've seen in cells, which is with an accelerated aging phenotype where there's more dysregulation earlier in life. Another thing we looked at here was our favorite biomarker, GDF15, to see a more continuous measure of energetic stress and how this correlates with allostatic load in our two groups. Again, what we see is that there's a significant correlation with GDF15 for the controls, but not so much for the mitochondrial disease participants. So having answered that, yes, there is higher allostatic load, we then looked at what systems this seems to come from, or what we can attribute this increase to. And what we can see here, as expected, is in the mitochondrial disease group, a greater percentage of the total allostatic load score comes from metabolic dysregulation. So there are more metabolic biomarkers dysregulated in mitochondrial disease than in healthy controls. We expect this to be true, and we see that this is the case. Interestingly, we see that hematologic dysregulation seems to contribute more for controls than for MitoD participants, and immune dysregulation as well. So these are the big differences we see between the two groups, but we also looked at differences within the two groups. So within people with mitochondrial disease who have already high levels of metabolic dysregulation, what distinguishes people with high or low allostatic load? So to do this, I did a PLSDA of people with high and low allostatic load in either the control or MitoD group. What you can see is up at the top are the factors that best distinguish people with high and low allostatic load among people with mitochondrial disease. And you can see a lot of those have the green, they're immune markers. So we have white blood cell count, and then we have specific cell subtypes as well that you heard about in previous sessions. And we also see norepinephrine distinguishes these groups quite well. In contrast, if you look closer to the bottom, you see factors that are important to distinguish controls with high or low allostatic load. Some of these, for example, insulin was also important among people with mitochondrial disease, but some, such as RDW there at the bottom, that's the distribution with the size of red blood cells, this seems to be important to distinguish between controls with high and low allostatic load. So it seems like maybe different factors within each group are contributing to the systemic dysregulation scores we see. Lastly, we then looked at how or if the allostatic load we see interacts with psychological stress and how this may differ between the two groups. So we did this by measuring psychological stress in a couple ways, which I want to point out to you because they seem to interact quite differently. So first, we used STRAIN, or the Stress and Adversity Inventory. Cynthia talked about this as well. This is a questionnaire that quantifies lifetime stressor exposure. So participants are shown over 100 different stressors that could be experienced across a wide domain, such as housing stressors, legal stressors, marital, et cetera. And for anything they say they've experienced, they're then asked about duration, severity, and other questions like this. This has a really broad conceptualization of stress, which is great. And in the end, we aggregated this into two metrics of stressor count and the feeling of threat experienced kind of cumulatively. Second, we use the Trier Inventory of Chronic Stress, which looks at chronic stress in specific life areas, specifically over the previous year. So participants are asked to rate the frequency and severity of specific stressors, mostly at work discontent, or being overextended at work, or feeling socially isolated, different metrics like this. Lastly, we use the Childhood Trauma Questionnaire. This measures specifically trauma experienced in childhood. So though I've brought up allostatic load as something related to repeated or chronic stressors, there are also times in life, sensitive periods, where one stressor or one severe stressor can have lasting effects, and childhood is one of those times. So to do this, participants are asked to say the frequency and duration, again, of specific traumas they experienced during their childhood. So what we see here is the Spearman R, so the correlation between the stress measure we're looking at, the top two are strain, and then the inventory of chronic stress, and allostatic load. In the first column, we see this for the entire group together, and then we see this split up by controls and mitochondrial disease participants. And as you can see, there is a much stronger and significant correlation between stress and allostatic load for people with mitochondrial disease, but we really do not see a strong correlation for controls. This was surprising and not what we expected to see. I also then looked at childhood trauma and how this relates. And here we see kind of the opposite pattern, where we see for controls who experience childhood maltreatment, there's about a 21% increase in the average allostatic load score, whereas for people with mitochondrial disease, this effect is absent. I want to be clear, this is not me saying that I think childhood trauma has no effect on dysregulation for people with mitochondrial disease, but it's just not what we see in these particular participants. Lastly, I looked at a measure kind of outside of the psychological realm of current professional class. And to see how this related to allostatic load for both groups. We see kind of a similar pattern where for controls in professional or managerial positions, these tend to be higher income, more control over the day, things like that. They tend to have a lower allostatic load score. Whereas for people with mitochondrial disease, there is a slight difference, but it's much smaller. So overall, what we saw as expected is allostatic load is higher in people with mitochondrial disease, and it correlates with age only for controls, not for people with mitochondrial disease. We also saw, as expected, system-specific dysregulation in mitochondrial disease, with them having more metabolic dysregulation. And then surprisingly, we saw that allostatic load was more strongly correlated with lifetime stress exposure for people with mitochondrial disease than for controls. So what this could mean, we see pretty clearly that there is some form of a link between energetic stress and multi-system dysregulation. We see that both with the genetic mitochondrial defect group having higher AL and for the correlation between GDF15 and AL for controls. What this could mean is that energetic stress itself could lead to allostatic load, or that allostatic load can incur an energetic stress. I think one thing that I found surprising and important to note is that even dysregulation arising from genetic mutation, so this metabolic dysregulation coming from mitochondrial defects, as we would expect, even this is not static. This is something that is altered over the lifespan, for example, by the amount of stress that people are experiencing. So I think this shows the need for interventions to provide support to decrease chronic stress for people with mitochondrial disease. Great. Thank you all for your time.